Good afternoon or good morning to all of you joining us from Europe, America, or Asia, or other parts of the world. Greetings from the sunny Goshen. I am Laila El-Hash, and I am the marketing manager at MSAID, and I'm happy to welcome you to our webinar on our latest release, the MSAID Platform. Please note that this webinar will be recorded for documentation purposes. And today we have an exciting agenda. To start off, Martin will provide a brief overview of the MSAID platform powered by CHIMERYS 4, our search algorithm. Following that, Daniel will present a live demo session covering a step-by-step guide on how you can analyze your data and also explore practical applications. And towards the end of each session, they will address all of your questions. And we encourage you to participate by asking questions and also sharing your thoughts in the Q&A session throughout the presentation. We will do our best to answer your questions during the Q&A sessions that follow. And with that being said, I will now hand over the virtual stage to Martin to begin his presentation.
All right, everyone. Can I get a thumbs up if you hear me and see the slides? Perfect. All right. Greetings also from my end, calling in from Cologne. I'm going to give you a brief overview of what the MSAID platform actually is. But before diving into the details there, maybe a couple of words to MSAID as a company. MSAID stands for Transforming Mass Spectrometry with AI and Aiding it in the Process. So MSAID, and our goal is to essentially take the heuristics that we've been applying for the past 25 years and substitute them with artificial intelligence powered by the vast amount of training data that is being deposited into the public domain. You might know us predominantly from our flagship product, and that is CHIMERYS, our intelligent search algorithm for bottom-up proteomics data. Just for those of you who are unaware, CHIMERYS performs the task of identifying peptides from mass spectra in a completely different way as traditional search engines. And in essence, it tries to explain as much experimental intensity, that is experimental MS2 fragment ion intensity, with as few peptides as possible. And it does so by using a linear combination of the predicted fragment ion intensities. And in the process, it can perform deconvolution of chimeric MS2 spectra, which essentially makes it capable of analyzing any type of bottom-up proteomics data with the very same concepts. Just briefly going over the workflow, we start with an intensity-based scoring, then produce a couple of PSMs, then we recalibrate our models, and later on perform the deconvolution as mentioned earlier, and then perform error estimation via the public tool Mockapod. CHIMERYS is now in its fourth iteration. We started out in 2022 with the release of the first version of CHIMERYS. Back then, it was integrated in Proteome Discoverer 3.0, and it could only process DDA data and was restricted to only methionine oxidation on when it comes to post-translational modifications. In 2023, we released CHIMERYS 2.0, which was the unifying approach. Back then, it was integrated in PD 3.1, and it could process for the first time DDA, DIA, and PRM data with the very same logic, as I mentioned earlier. And on the PTM side, because of advancements on our prediction model in Ferris, we were able to support additionally also phosphorylation, and PTM localization was handled in PD 3.1 with PTMRS. And this year at ASMS, we released CHIMERYS 4.0. This will be integrated in Proteome Discoverer 3.2. It can also process all different acquisition methods, and now also add support for both acetylation and ubiquitination, as well as a native PTM localization algorithm that makes use of predicted intensity and can calculate probabilities of where a PTM might be located in a peptide sequence. Just to give you a brief background on what you can expect when you use CHIMERYS for your data analysis of acetylated and ubiquitinated data sets. Here we compare the performance of CHIMERYS versus in orange versus sequenced HT in blue. And in the plot on the left, we have acetylation, and the plot on the right, ubiquitination. And then the groups of bar charts are always PSMs or peptides. And as you can see clearly, the gains that you can obtain when you use CHIMERYS as opposed to sequenced are substantial, in the order of 50 to 70% more peptides. We are planning to effectively support more and more PTMs as we go along. So stay tuned for that. However, as I mentioned earlier, prior to the CHIMERYS 4 release, localization was restricted to PTMRS. That worked fairly well on DDA data. So here is a precision recall plot comparing the PTM localization on a known data set as performed with either CHIMERYS 2.0 and PTMRS in light blue, MaxQuant in orange, or CHIMERYS 4.0 with its native localization algorithm in dark blue. And as you can see, if you are not used to reading these plots, you want to maximize the area under the curve here. So you want to get the most recall at the best precision, and vice versa. You see that effectively CHIMERYS 4 performs much better with its native localization than CHIMERYS 2 and PTMRS, and even than MaxQuant on this DDA data set. But of course, people are more and more making use of DIA data. And if you look at the performance of our localization algorithm on DIA data, what you can see immediately is that the new algorithm outperforms the localization calculation by Diane, but it also outperforms CHIMERYS 2.0 with PTMRS, which in fact didn't work really at all on DIA data. So if you're planning to do PTM analysis with DIA, we highly suggest you move to CHIMERYS 4 and its native localization. Why am I telling you all of this? Well, of course, CHIMERYS 4 is the next big step in proteomics data analysis. It is AI-powered software at your fingertips. And with its integration into Proteome Discoverer 3.2, you will be able to access it from your Windows PC. But you wouldn't be here if you wouldn't be also interested in the second way of being able to access CHIMERYS 4, and that is via our all-new MSAID platform powered by CHIMERYS 4. It is available right now, and I'm going to tell you all about it in the next couple of slides. But before I dive deep into what the MSAID platform actually does and you know how we built it, let's take a step back and just think a bit about where we are as a field of bottom-up proteomics, and you know what the big advancements were over the last couple of years. We have seen tremendous advancements on the side of the machines. The machines are getting faster. Chromatography gets better. We get better peak capacities on the new columns. People are moving to very interesting new column setups with very interesting new chromatography apparatuses. And on the measurement side, we made big strides. But on this data analysis side, what we have identified is that there is a certain lack of automated pipelines, and that effectively results in very manual workflows that are governed by repetitive steps. And those are prone to error because they are very manual in the majority of all cases. And very often we see disconnected patchworks of local infrastructure. And this is not something that we made up. So here's a little story that I've experienced myself when I was still working in the wet lab. Back then, we used to acquire our data, and the data was ending up at the instrument acquisition computer. And from there, we transferred it via an external hard drive to the processing PC on that processing PC, we did our data analysis, but the result files couldn't be stored permanently there. This was a shared resource. Everyone needed to use that computer, so those needed to move away and ended up in everyone's home folder. For the person working on the project to find it later, but as soon as that person moves to the next position, it was always hard to take over old projects. In the past, that has been tackled to some degree. One solution here is, of course, the RDI workflow as presented by Thermo Fisher, which basically ties the first 3 steps already together. You have the data acquisition, you have the storage of the raw files, and the processing very tightly coupled via the RDI ecosystem. But the storage and the long-term storage and integrative analysis of the results is still lacking to some degree. It is still a very manual process and very cumbersome to do systematically. So result storage is something that we wanted to improve, and so did we want to improve the data analysis and make it possible for people without computational knowledge to actually analyze the vast proteomics data sets that we encounter nowadays in order to end up at the actions that everyone wants to do. And in my case, when I was in the lab, result storage, as I mentioned, was in the home folders. Data analysis was either done with Excel or GraphPad Prism, if people had access to that, or R if you have the knowledge to perform these analyses. But very often the bioinformaticians that will help you with such analyses are actually not available, are bound in other projects. So we identified this as a risk to actually get to the actions that are embedded in your data as quickly as possible. So with these challenges identified, we set out to develop what we call the MSAID platform. Don't be alarmed by the relatively generic name. We were very hard working on the platform to get it ready for ASMS, so not much time to come up with a very inventive name for it. But I'm sure you'll appreciate in the next couple of slides that it's really a platform aimed at serving as one ecosystem from raw data all the way to insight. So what are its key features? Well, it's an all-in-one cloud solution, and it's accessible from your laptop and from your phone. So if you're the type of person that would, on the sofa on a Saturday or Sunday evening, watching Tatort, as we do in Germany, start another search of some raw data that was acquired over the weekend, that is certainly possible in the comfort on your own couch from your phone. So given that it's a cloud platform, it seamlessly scales with data volume. It has elastic parallel cloud compute. So if you have a lot of data coming in, you very easily can scale out the analysis of that data to multiple different servers and expedite the generation of results. It features systematic result storage in the form of a data lake where we deposit raw data to be looked at later on and by multiple people collaboratively. For that collaboration, we provide flexible data interrogation, so you can access the data via an API. You can access the data via a data browser, but also via visualizations that are pre-built, and I'm going to show you a couple of those in the next slides to come. But the API that I mentioned earlier that you can use to access the data can also be used to fully automate the platform. So if you want to integrate the MSAID platform into your workflow, that is certainly possible because it has a full-featured API associated with it. And all of this comes with a subscription-based model being hosted on AWS. So let's briefly go a bit more in detail about over the key features. One key feature is that we a dedicated user space right in your web browser where you can deposit your data either from your web browser or via a CLI client directly from your acquisition computer. So these are the two methods of effectively uploading data. And we envision certainly the CLI client being very useful in the future for automated workflows. So once the raw file has finished acquisition on the acquisition computer, it can immediately be uploaded to the MSAID platform. And if you want to, even immediately searched with a custom set of settings that is applicable to that particular raw file. In the future, we were thinking about making browsers so FTP-based browsers like WinSCP also available for upload. But we believe that the browser-based interface and the command line client are already two very good tools in order to get the data into the cloud as quickly as possible. Once the data is in the cloud, you can manage it and organize it with a Google Mail type tag system. And what do I mean by this? Effectively, every single raw file, its search results and samples that are contained within can be tagged with free-form text. What does that mean? So for example, here, if we zoom in, we see that the first raw file is associated to a particular project. That is the name of the publication. It comes from a certain PXD number, and it contains ubiquitinated peptides. And by using these tags and adding as many tags as you want to the raw data, you'll be able to find back old results and make use of old results in the context of newly generated results as well. We envision that in the future, this manual tagging that you can do right now will be supplemented with automatic tagging as well. So we envision extracting automatically from the raw file meta information such as, for example, the type of analyzer this was run on, maybe certain quality control metrics that you can think of, maybe the collision energy that was used, or the collision method that was used, so HCD or CID. Integration into LIMS system is always planned for the future, but for now, the tag system is already a full-fledged system that you can use to easily retrieve and categorize your data. As you can see here, the system is set up in a way that you can reuse old tags that you have created previously. So let's say you have an experiment with a couple of hundred raw files. We make sure that when you tag a raw file with a specific tag, say, for example, a PXD number, then that PXD number is persisted in the database and can actually be retrieved, so you don't have mismatches of slightly differently spelled tags. So effectively this way, you can build up your own controlled vocabulary in order to access your raw data and the results of it. I mentioned earlier, the platform is powered by CHIMERYS 4, but it is designed to support multiple algorithms. We are now in the process of contacting various different developers of open source tools for the analysis of bottom-up proteomics data in order to see whether they would be interested in integrating their software also in our platform. Because for us, that's fairly straightforward to do. All we need to do is write the adapters for the input data and the corresponding output data to bring them into the same format. So if you're a software developer, feel free to approach us and we can see how we collaborate. And I think there's big value in such a collaboration because it enables you to scale your data analysis in the cloud to vast numbers of computers. Here, I brought you a little example of an experiment consisting of 1 to up to 100 raw files. And what we're looking at here in this bar plot is the time it takes to process one of these raw files or the entire experiment of 100 raw files from start to finish. And you can see that processing one raw file takes roughly 52 minutes, while processing 100 raw files only takes roughly 78 minutes, which is a bit more than a 50% increase in runtime for a 100-fold increase in the number of raw files that you've actually searched. So it scales out very nicely and will allow you to search through very big experiments with ease. As the data is searched, you can explore information on the experiment. You get a live view of the logs of the software that is currently searching your data. And once the experiment was successfully completed, you get stats on, say, the number of PSMs or the number of peptides for that particular search. We mentioned here searching 100 raw files in one experiment, but we think a very powerful addition to that is the possibility in the platform to actually combine experiments. What does that mean? It means that you can search your raw files one by one. For example, a raw file was acquired, immediately is uploaded to the MSAID platform, and then immediately processed individually. You can later on combine the search results of these raw files and generate a new experiment where we only consistently calculate peptide FDR, protein group FDR, and quantification on that set of data, allowing you to bypass the need to research your data once the entire experiment was acquired. And that saves a big amount of time, especially with big projects, and allows you to continuously acquire and analyze the data as it comes in, perform quality control on it, and don't have to research the data once the experiment is completed. For an initial glance at the data, you can use the data browser, which is effectively a bare bones implementation of Excel in the browser, if you pardon my French. Effectively, what you can do is look at your favorite protein. Is it there contained in your data set? How many peptides do you have in your data set for that protein? And how many PSMs you have for that particular peptide. So you can do a bit of high-level analytics on these tables, and it's blazingly fast. But of course, exploring bottom-up proteomics data, based on tables is very cumbersome. So we also added capabilities to effectively visualize your data with standard plots such as bar plots and scatter plots. And that set of plots will, of course, be expanded with future releases of the platform. And those plots will then allow you to get insight into your data. But if that is not sufficient, you can always download your data and analyze it in your favorite tool using either R, Python, Excel, or whatever you're used to. That download capability is a central feature of the platform as well. Right. And with that, I'm basically at the end of the presentation. All that remains to be said is if you're interested in the MSAID platform, please try it for yourself. You can sign up today on the platform at platform.msaid.io. Generate an account. We will gradually admit people to the platform as they come in. And you will be able to demo the platform for 1 month for free and process up to 100 gigabytes worth of raw data in that month so that you can really evaluate whether the platform brings a measurable benefit to your workflow. So yeah, all that remains to be said is thank you very much for your attention. And if you have any questions, feel free to shoot. Thank you very much.
Thank you very much, Martin, for your presentation. And I see 2 questions in the chat on PD 3.0 and also proteomics workflow. So I'll hand it over to you, Martin, so that you can begin by addressing the questions.
You're talking about the Q&A?
Yes, we have
yes,
one in the
Q&A.
chat and
okay,
the other in the Q&A session.
All right, so let's start with the Q&A one. The question is, B1 ion information and detection is a problem. and N-terminal acetylation is being used to detect and improve the quality of MSMS and improve de novo sequence efficiency. We'll be happy to use the platform. Can it be used on a PC? So effectively, you can use the platform on a PC because it's browser-based. So there's no problem with you just opening Chrome or Firefox and navigating to the platform and then trying it out. We have the capability to do acetylation. Feel free to reach out to us if you have any trouble setting up your specific experiment. But that certainly should be possible to analyze on the platform, and should well fit into the 100 gigabytes that you can process in the first demo. I hope that answered the question. Then let's go to the chat. When will PD 3.2 be released? I cannot comment on this, unfortunately. So if you're interested in PD 3.2, I'd suggest that you reach out to your thermal sales representative in order to get more in-depth information about the release dates of PD 3.2. Do we have any other questions at the moment? I don't see any in the Q&A section and none in the chat. There's one person has their hand raised. Can we unmute the person to allow asking the question?
I just did. I think you should be able to ask the question now.
You just need to unmute yourself, I believe. So we allowed you to speak. You need to just unmute you in the top right corner next to the share button, I believe it should be. In the meantime, can you talk briefly about the subscription scheme? I believe Daniel will go a bit more into detail on this topic. I'd suggest to hold off for his live demo, and he'll give you some information on the subscription. Professor Jagannandam, sorry if I might have pronounced your name incorrectly. I think the easiest would be if you can just paste your question into the chat, and then we can answer it. Oh, can you comment on compute cost, platform compatibility, and data privacy? Question from the audience. So compute cost, again, that is associated with the pricing scheme and the different subscription tiers that we have. Daniel will go a bit more into detail there. For now, the platform is compatible with thermal instruments, but we explore compatibility with other vendors as well as we speak. And data privacy, we are in the process of setting up an ISMS and starting ISO 27001 certification. While this is not available at the moment, we operate the platform to the highest privacy standards and use industry-leading technologies in order to protect the user's privacy. If you have specific questions as to privacy of raw data or privacy of results, feel free to reach out and we can set up a dedicated meeting as well.
Thank you very much, Martin. I guess, and now we will proceed to the second part of the webinar, where Daniel will present a live demo session. I see your screen, so welcome, Daniel. The stage is yours.
Yeah, thank you very much. And thank you everyone for being here. Fingers crossed, live demos are not what people like to do when developing software. But since this is a product that is already available, we will take that risk. So I will give you a brief glimpse into the interactivity of the platform that Martin just described, showing you what you can see on it and maybe expanding into one or two areas that Martin has skipped so far. So as Martin initially said, you can go to platform.msaid.io and register an account, and we will admit you to the system to test the platform. The platform is hosted on AWS at the moment. We are currently storing all the data that is on that platform in the European region. We might add additional regions for data availability later on. The data of European customers will always be in the European region in order to comply with GDPR. So when you log into this platform, this is the dashboard that greets you, and you might have seen before. And the goal that we had here was to try to bring the proteomics scientist something that he hasn't been used to, and that is user experience, so good UX, good UI. Because we use a lot of tools that are command line based, we use a lot of tools that have been developed by academic groups, and sometimes they have great UI and have a great UX, but a lot of the times we're just running stuff in our command line. And we really strive to create something that is very intuitive to even people that have not yet touched a proteomic search engine so far. So when you log into this screen, what you will see is a overview over your account. This is my account. I have uploaded 34 raw files. I've uploaded FASTA files. I've created experiments. But I want to show you how seamlessly this interaction works. So you can basically go and organize your files right in your browser, which are deposited in a user-specific S3 bucket that nobody else has access to. And you can monitor both the upload and the download of these files just in your browser. What you see is, as you know, for every proteomic search, you need a FASTA file, you need a raw file. So let's make sure that we quickly upload a raw file and a FASTA file. And what we put a lot of effort on is making sure these interactions are fast. Proteomics data is getting bigger and bigger, and people don't like to wait. Unfortunately, cloud-based processing means you need to upload your data at least once. So we had to make sure that this data upload is fast even in a browser. We have benchmarked this on servers sitting on a 1 gigabit connection in order to mimic a fast industry internet. And we have achieved speeds that basically cap out at the speed of the internet connection, meaning you can pump data into that S3 bucket at the speed of 100 megabytes per second, or close to 1 gigabyte bit per second. You will see that the data uploaded here is implemented in a tabular design. As Martin said, you can add text to this. I will just add the tag webinar. And I can't write, so now I have the tag Webby. And you can see that you can organize them. If there are multiple people in my organization, I can look for my files, and I can also sort and filter all of these data depending on how I want it. For example, by just clicking on tags and finding back data that we need. Right, so now after we uploaded a raw file, let's upload a FASTA file. This is a little more fun because it's a little bigger. So we'll upload a FASTA file here. We can watch it upload. We can also watch it upload here. And that takes a couple of seconds to upload. So I'll already tag it really quick. As my data is now merging, we can go into starting to create our own experiments. Just as your files, which are organized in a tabular fashion, also your experiments that you have already run, organized in a tabular fashion. Again, I have a complete list of raw of experiments that I've run the last couple of days. I can tag them, I can order them, and I can look at details of them. But before we go into details, let's first start an experiment here. And again, we put a lot of effort into creating a simple but comprehensive wizard that will guide you through the step of setting up an experiment. So you make sure that you have sensible defaults and don't have to put in information more than required. So let's create an experiment here that we call webinar for all I care. We can again ask a couple of tags. I can also scroll my last couple of tags that I have. I will add a HeLa file, so I'm just going to give it a TILA file, and I can select my algorithm, which in this case is CHIMERYS. And then I have to do the inevitable. I have to select FASTA files. I have to select my raw files. Here's my example 7-minute raw file. I can also filter for that. I can select this file. I can again look if all the additional information is correct, and I can just go into the next step. Here, I have the ability to select my settings for my search engine. And because I don't want to do this a lot of times, I can template that in a way that I can save my favorite settings for my search engine. So creating an experiment is basically 5 clicks. 1 click opening the wizard, 1 click selecting a FASTA file, 1 click selecting a raw file, and then 1 click submitting the search to the system. I can select here a couple ones. So I can just load mine, which then will give me my regular settings. You select an enzyme, you select your peptide length, you select your modifications that you want to search with. All of that is known to you if you use proteomic search engines. And unfortunately, we have to do this once. In this case, it's pretty boring. It's just a HeLa file that I'm going to search with carbamide methylation and oxidation. In a next step, we can select different types of quantification. CHIMERYS at the moment allows quantification on MS1 level for DDA, on MS2 level for DIA, and on TMT level for MS2-based reporter ion quantifications. In a last step, I will then see a summary, and I have the ability to apply this template and send it. Because even that would be a bit of too much time, I created a couple of experiments already for us to look at. So how would that look like if I have an experiment which is currently running? And I can monitor these either on the web, I can also monitor these on my phone, I can monitor these on my laptop or iPad if I want to. So what you will see is that running experiments will have a detail view where you can look at the logs, you can browse all the logs that have been created by the service, seeing where all your individual files are being processed as we go along. You can also see what kind of system files you submitted to the system, and you can also see what kind of settings you used to create this file. In the new version that we're deploying this week, you'll also be able to download this and store this away, since this has been a popular request that people like to store away a PDF file together with their experiment so they know how they actually run this. What we also can see is they can already have a quick glimpse into the data, where you can see the number of proteins, number of peptides, number of PSMs in that, which can be used for QC purposes or just to check whether the run was as successful as you wanted it to be. If you would like to download the data, which is obviously the most boring way to interact with data on an online platform, you can basically just request or download, select how your data should be filtered, select which kind of tables you would like to have, maybe save a little space here and remove the PSMs, and wait for the download to be created. What's happening is that back in our S3 bucket, which is a scaling infrastructure to store a ton of files, a worker is now going and collecting the data for you that you then can download once it's been prepared for you. But now let's look at a couple of features in these experiments if you want to explore your data on the web. And as we said, this is our very first initial release, and we'll build these tools, but still, this is very neat to interact with. So first, let's filter our list for data that I wanted to show you, here indicated by the webinar tag, and look at the data browser, which you can just do by looking at the individual protein groups, peptides, modified peptides in precursors identified in that sample. If I open my protein groups here, I can also look at PSMs which are associated with that. And obviously, I have a full range of filters available to filter my data to my needs. What is also coming in this release next week is going to be the option to properly parse the FASTA headers, which are at the moment not very cool to look at, but parse them into taxonomy IDs, protein identifiers, organisms, and gene names, such that you can more easily browse your data and figure out if, in this case, your keratin is really there. If we go back, the most important part of data exploration is obviously visualization. And why we might not be there from what kind of plots we already offer, we believe this is going to grow into something that can aid you with both quality control, but also in-depth exploration of your data. If we now go to the data browser here and to our visualization dashboard, you will see that every experiment has a visualization dashboard, which is stored, which can be modified, which is interactive, which can be reordered the way we want, which can be used to export the data, export the plot, which can be used to change the plot, and obviously it can be used to create new plots that would help you explore the data. As you can see, at the moment, bar plots and scatter plots are available, and we're working hard on adding all of the important plots that you have been using in your R session, in your Python notebook to explore the data. Again, we created a very simple wizard that can basically, on the click of a button and with as little settings as possible, create a new plot about, in this case, the precursors in every single file. The plot here was added to the bottom because it's the last plot we added, and this is interactive. You can zoom it, and you have hover over for all of these raw files that you would like to look at. In this case, I searched 2 pretty big HeLa files together with a couple of small files to illustrate an effect here. Another feature that we're going to add soon is downloading the data completely from these dashboards. The dashboards right now can already be shared between people that are in the same organization and can be viewed as such. We also envision to add dashboards for quality control purposes where you're not comparing samples within an experiment, but where you're comparing experiments, for example, on a longitudinal scale. For example, your HeLa files that you run, hopefully, frequently in your day-to-day operation, and then can see how your identifications, how your quality metrics will change over time. We are also in the final testing of a CLI client that can interact with the platform if you would like to apply, upload data, start searches, or at some point also download searches using a command line interface or a non-browser interaction. And we believe that this will make the platform even more usable for people that actually have skill sets to automate these kind of processes. We have started writing extensive FAQs to all the functionality on the platform. So if you register yourself and want to give it a spin, I guess this is a pretty good place to start. We've also produced a product tour which will recap a lot of these points that I have just shown you here in this session. One point that we haven't talked yet about is the available resources and how we bill and how we manage our subscriptions here. So on the right-hand side, you will see that in this case, I have approximately 1,600 gigabytes of storage left and 1,400 gigabytes of compute left. And this is exactly how the system is priced at the moment. Currently, we bill people for the amount of data they submit to the system and the amount of storage they use. This is organized in something in tiers where we have different already pre-made tiers that fit your needs. We have a starter plan that should have a very low entry barrier to start exploring the platform and process data on a time-to-time basis. We also have a premium plan that is sufficient to support any non-astral mass spectrometer, including all files for the full year. However, as Martin said, the cloud itself is scalable. That also means that your tier, the compute that you can have, and the gigabytes that you can have is flexible. That means that depending on your use case, we can create a quote and an account that would match the needs of your organization. I don't want to go too deep into pricing. The only thing that I want to say here is that the initial tiers are available from 400 euros a month, which we believe is a very low entry barrier into the world of cloud-based proteomic data processing. If you want to know more, log into your account, just request a quote, give us a little information about you and yourself, where you're located, what your profession is, and we will sit down together with you to figure out what kind of plan would fit your needs and how we can get you onboarded to the platform as soon as possible. With that, I would like to close our little tour of the platform and be open for more questions to be asked. And I'm very happy to hear thoughts. Overall, I can again invite you to please make an account, figure it out, or sit down with either Martin or me to discuss what kind of features that might have not been in this webinar are missing for you. A SaaS product is an ever-growing and ever-changing and ever-improving system, which means that whatever you're missing right now might be just an update or two away. So please don't be shy on requesting compatibility that we have not yet demonstrated so far. We are very happy to sit down with you and discuss about that. And with that, I am open to questions.
Thank you, Daniel, for the live demo of the MSAID platform, and also for answering the previous questions on compute cost, compatibility, and also the subscription scheme. I see two questions in the chat related to FASTA files, FDR calculations. So I will hand it over to you so you can begin addressing the questions.
Absolutely. And I'm also realizing that I missed a little part that I wanted to talk about here in this demonstration, which is user management. So at the moment, we have implemented a rudimentary user management where we have accounts which are sub-users of another account, which can collaborate on the data itself. The idea here is that you have a daemon account or a general account for your lab or for your organization, and you can basically invite other users to join you on that journey. You will see that in this case, I have two more sub-users here, one of which I've deactivated. I can see exactly what these people are doing in their accounts, how much compute they have used, and what else is going on. Inside one of these organizations, you can see experiments, you can see files, and you can use experiments and files to work together collaboratively. Only the owner can delete stuff that you don't own. However, this is perfect, for example, sending out the dashboard or the link to the data browser to your colleague and telling him to have another look at that. We will also be expanding this collaborative data exploration if we feel that people like this feature a lot. I personally was always very happy if I could quickly share data that I had instead of making screenshots and sending that via Teams message.
Awesome. Thank you very much, Daniel. So let's proceed with the Q&A, all the questions that we have on the Q&A section.
All right, let me switch over. I see quite a lot of questions, and now let's make sure that we don't miss anything. Okay, so let me start off with the points that are concerning the commercialization again. The compute cost, I think I covered. We can definitely sit together and work out a tier. As I said, the minimal compute cost for the smaller tiers are somewhere in the realm of 400 euros a month. Depending on how much compute you would buy, we'll obviously also offer rebates in terms of how much the individual gigabyte would cost you.
Maybe I can pick up a question, and that is the question of sort of reducing the search space to tailor it only to peptides of interest. Here, one example was the high abundant plasma proteome, so only 4,000 to 5,000 proteins instead of using a whole human FASTA file when analyzing a plasma proteome data set. This is always a bit of a double-edged sword. Of course, the sort of general approach taken by most peptide-centric search engines is to generate a spectral library that is highly specific to your sample of interest, or generate that library on the fly. And there, this restriction of the search space is already baked in somewhat in the search process. I would think you can certainly try, as long as the number of proteins that are in your FASTA file doesn't drop below a certain threshold. Luckily for you, if you're worried about FDR control, there's a way to measure whether it adversely affects FDR, and that is to perform entrapment experiments. If you're interested in the theory behind entrapment experiments, I encourage you to take a look at our YouTube channel, because there we have a couple of videos already online that go a bit into detail as to what an entrapment experiment is. Just one sentence on it, it effectively allows you to validate whether the FDR control of a search engine is performing as expected, and gives you the FDR that you've actually set in your workflow. So I would say give it a try. But make sure that if you use this in production, that you carry out entrapment experiments to make sure you don't fool yourself.
Thanks, Martin. I'm going to answer a question by Marcus, who asked if we are planning to integrate a biology layer onto the platform, whether it's going to be restricted to data QC. So the way we see this is that what we just have shown you right now is the beginning. So what the platform essentially is, it's a systematic queryable storage of result data on the cloud. And obviously, one of the goals is to integrate that with the annotation services to allow you to better understand what's going on in your data and what the biology is. It won't be tomorrow, but we're definitely planning on working on themes where you can annotate your data using Go terms, where you can annotate your data using external data sets. I'm also imagining interactivity with other platforms where we have already stored a lot of data and try to reprocess that in order to contrast your data. So yes, definitely the biology angle is something we want to follow up here. What we have created is the core element, and now comes the fun part in a science way.
Yeah, let me pick up another question, Daniel. There's the question whether the result of spectra, so PSMs, peptides, and MS1 and MS2 XICs are available. So when it comes to PSMs and peptides, what Daniel has showed you in the live demo is which PSMs were identified for which peptide, what their Q value is, etc., etc. At the moment, there's no functionality to visualize the corresponding spectrum with a mirror plot, and there's also no functionality to look at XICs. However, we believe that the classical way of looking at XICs with the fragment ion traces along retention time can be supplemented with another visualization that we are in the process of developing, and that is our CHIMERYS coefficient along retention time, which is effectively an interference-corrected total ion current for the matched fragment ions, and it resembles a summed XIC without interferences along retention time. This is something that is within arm's reach, so to speak, but we are already in the process of investigating whether what changes would be required in order to also visualize XICs on the platform.
Thanks, Martin. Another question here is on the compatibility of SILAC and dimethyl labeling and tandem labeling. So I can tell you that the platform already can handle SILAC and tandem mass tags as labeling reagents. Dimethyl labeling is something where we do not yet have the compatibility to, but if this is something that's really interesting to you, we can definitely add that to our feature list, as dimethyl labels are quite ubiquitous and it should be doable to find training data on this. The second part was if we can download and save the analysis scheme. So yes, this is definitely going to be possible, that you can basically save your experimental design and setup that you used to create this experiment, and that this is downloaded with every of the experiments, so you can basically keep this as a record or make use of it to put this into a new experiment that you're creating. One little side aspect to that, we are also planning to release a bigger release where you can tell the platform your experimental design, meaning that you specify which TMG channels or which sample, which raw files or which fractions belong to which sample. And we already in the background can do calculations for you that you would usually do during your data post-processing. This is going to be also on the platform in the next couple of weeks. The implementation of that is nearly ready. And that now then also renders the visualization functionality much more useful, in a sense that if you can already tell us which are your controls and which are your treatments, we can already pre-fill your wizard to create a volcano plot, for example. And this is then where this optimization on the web starts to be in full effect and starts to give you a benefit that you don't have if you do stuff offline.
Thanks, Daniel, for the nice insight. Maybe let me pick up another one here. Maybe we haven't talked about this yet. So the interaction between CHIMERYS and PD seems to be pegged at 48 hours runtime. So I believe what you are referring to is that you can at most get 48 hours of processing time per 24 hours of compute time. At the moment on the platform, there are no such limits. So Daniel has been searching 100 raw files, and these 100 raw files have been searched in parallel. In the future, we might introduce limits, as we have learned that also AWS can eventually run out of computers to deploy for us. But at the moment, this is effectively scaling as needed. So there you can expect a much higher parallelism than is available currently as part of Proteome Discoverer.
And that, I guess we're slowly but surely coming to an end. I will answer the last question that here is in the chat, which is, can you create a spectral library for DIA files in the platform and use it for downstream processing? And the answer to that is not at the moment. Technically, we would have everything in place to do that. In case it's not clear, CHIMERYS can process DDA, DIA, and PRM data without you having to set anything. So DIA data analysis can be performed on the platform. If you would like to have a calibrated spectral library, and this is something that is dear to your heart, I would be happy to sit down and see what your requirements are, what kind of tool you would like to use that with, because technically this is definitely a possibility. The question is whether there is enough people voting for such an issue, and I'd be happy to follow up on this. And I guess with that, I'll hand back to Laila, I guess, because we're a bit over time. Thank you so much for all the questions. If you have anything else, Martin's email address is in the chat in 5 seconds, and my email address is as well. Feel free to reach out via email. We are happy to sit down and discuss any of your concerns that you might have or any of the features that you might require. Also to learn what the next talks should be about, what the next FAQ section should be about, and where we need to bring and shed more light to that.
Yes. Thank you very much, Daniel, and thank you very much, Martin, for the presentation and also the live demo. So yes, we're coming to the end of the webinar, and I just want to encourage you, if you haven't tried the MSAID platform yet, just scan the QR code or please visit us at msaid.de. And thank you for being part of our webinar. Thank you, Martin and Daniel. And we will ensure the recording is available on our YouTube channel for your convenience. Stay tuned and for more exciting updates from MSAID. And thank you very much. Have a wonderful day.